Hello, this is an informational video about the disease familial adenomatosis polyposis, better known as FAP. In this video, we'll discuss the general information about the disease. Then, we'll talk about the healthy gut, and after we'll discuss the mechanisms and mutations that cause the disease in the colon. At the end of the video, the treatment options will be explored. So, let's start! FAP is a disease of the bowel. It is an autosomal dominant inherited condition in which 100 to 1000 polyps form in the colon. Polyps are mushroom-like structures on the gut lining. While these polyps start out benign, they can transform into cancerous cells when they are left untreated. Worldwide, 7 to 10 out of 100,000 people are diagnosed with this disease, and it is estimated that it occurs in 1 out of 11,000 live births annually. Polyps usually don't cause any symptoms, but because there are a lot of polyps in FAP, the risk of getting complaints is bigger since they get in the way of the normal digestion system. Patients often go to the doctor because of non-specific symptoms, such as unexplained rectal bleeding, diarrhea, blood in the stool, abdominal pain, or because of excessive tiredness. The tiredness can be explained because of the high risk of anemia. This is because of two reasons. Because of the shearing of the content of the lumen against the polyps cause blood loss. And because of the polyps, the absorption function of the colon is disrupted and less iron than normal is absorbed. Other symptoms these patients might experience are bad eyesight from birth, changes of skin such as little bulbs, missing teeth, and sometimes benign bone tumors. Patients are usually diagnosed via a colonoscopy, and when the patient has more than 100 polyps, it is likely to be FAP. The initial assessment for the diagnosis is the collection of family history of cancers and premalignant gastrointestinal conditions. However, the diagnosis can only be confirmed by a DNA test. The material taken for the test can be a mucosal sample from the mouth, or a polyp sample taken by a colonoscopy. The prognosis of FAP is dependent on the cancerous state of the polyps. The risk of developing cancer is 100%, which makes this a very dangerous disease. To observe the malignant development of the polyps, the patients have a colonoscopy every 1 to 2 years from the age of 10 years old. To understand more about the cause of FAP, a good understanding of the microscopic anatomy of the colon is necessary. Let's start off with the global anatomy of the digestive system. When you take a bite of your food, it gets transported to the stomach via the esophagus. After being mixed with acid for a short while, the food leaves the stomach and enters the duodenum. Here is where digestion really starts through the release of enzymes from the pancreas. Whilst moving into the small intestine, which are the ileum and jejunum, it is being cleaved into small fragments. These fragments are selectively taken up in a process that is generally referred to as absorption. What is still left enters the colon, where bacteria seem to play a vital role in the modification of the nutrients and where water is taken up. In FAP, the colon malfunctions, so let's zoom into that. The first layer of the colon is the mucosa. This layer forms the barrier between the intestinal lumen and the internal environment. The mucosa of the colon is characterized by crypts. These crypts of Lieberkühn are little notches in the epithelium, so the surface of the colon increases and more absorption can occur. If we zoom in on the epithelial layer, it contains a lot of different cells. Most of the cells are enterocytes. They are located high in the crypts and their primary function is nutrient absorption. Then, you also have enteroendocrine cells. The enteroendocrine population of the large bowel is less diverse than in the small intestine. In the colon, most enteroendocrine cells produce the neurotransmitter serotonin. Goblet cells are rarer than enterocytes. They secrete mucus that protects the epithelium against luminal content. Another cell that protects the epithelium is the Paneth cell. They do this by secreting antibacterial proteins. These cells live at the base of the crypts and are part of the innate immune system. They live longer than the other epithelial cells and are important for the renewal of the epithelial layer. You can see there are also other types of cells in the crypt. Another type of cell is the crypt-based columnar cell, which is a stem cell. These cells are located in the stem cell niche, which is in the base of the crypts, and are alternated by the Paneth cells. The cells have a special marker, a G protein-coupled receptor called LGR5. There isn't a clear definition of a stem cell, but in this video we'll use the functional definition of a stem cell as a cell that, with appropriate niche factors, is able to reconstitute the tissue of its origin. The stem cell is responsible for cell renewal. This means that every type of cell in the intestinal epithelium originates from the crypt-based columnar cell. But how does that work? At this moment, there isn't a proven theory about the different processes that keep maintaining the stem cell niche. But there is a theory that involves an association between the Paneth cells and the stem cells that we'll explain right now. A stem cell divides every 20 hours. By this division, two aquipotent daughter cells arise. The fate of the daughter cells is sealed by the Paneth cells. One of the daughter cells remains a stem cell. This is the one that is in direct contact with the Paneth cell. The Paneth cells hold their neighboring cell in a stem cell condition by producing different factors. One of those factors is Wnt3, a protein which is part of the Wnt gene family, which we'll discuss later on. Another protein is R-spondin-1. R-spondin boosts the canonical Wnt pathway. R-spondin binds to LGR5. This reinforces the Wnt signal. This leads to more juxtacrine signaling from the Paneth cells to its neighbor and reinforces the stem cell factors which keep the cells adjacent to the Paneth cells at the base of the crypt. The reason why the Paneth cells don't migrate is not totally clear yet. They express epfrin B3. This factor is probably required to enable the positioning of the Paneth cells. The other daughter cell differentiates in the committed progenitor or a transit amplifying cell. Only when the committed progenitor cells have switched off the canonical Wnt pathway, the cell cycle will arrest and they can escape repulsion. Now, it can leave the stem cell niche. It is then located somewhere halfway the crypt. Here, the cell will differentiate in one of the different cells present in the colon epithelium. After 4 to 5 days, the cell has migrated to the upper part of the crypt where it will undergo apoptosis and will shed into the lumen of the colon. This short lifespan of the enterocyte is the main reason for the need of cell renewal. But let's go back to the epithelium. Epithelia is a tight sheet of cells that forms boundaries between two distinct anatomical structures. In the gut, they form the boundary between the outside and the inside of our body. So, it is important that there's a tight connection between the cells of the epithelium. This connection is made by different junctions. The first junction is the tight junction, also called the zonula occludens. This junction provides real protection against the content of the lumen. It also maintains the polarity and the transcellular transport of the cell. The tight junctions are made out of two proteins, occludin and claudin. The next junction is the adherence junction or the zonula adherens. The junction is flexible and enables the tissue to contract and relax, so there has to be a connection between the neighboring cells. Both the neighboring cells have E-catenin, a transmembrane protein, that are connected to each other. This connection is stabilized by calcium, which is shown in this figure. Caterins owe their name to the finding that they require calcium for proper functioning, calcium-dependent adherence. The smaller intracellular part of caterin will bind to P120-catenin, which is bound to beta-catenin. The beta-catenin is bound to alpha-catenin, which is a linking protein. The alpha-catenin is bound to the microfilament F-actin, which is a linear polymer and part of the cytoskeleton. The F-actin microfilaments are held together by alpha-actinin. Later on in this video, we'll explain the process of this construction in more detail. Now, let's take a look at the processes at cellular level. Right now, there are thousands of signaling pathways active in every cell of your body to keep it alive and responsive to the needs of the organism. One of those pathways is the canonical Wnt-signalling pathway, which is designed to activate gene transcription in the nucleus. The important protein in the Wnt pathway is beta-catenin. Beta-catenin is a dual-functioning protein involved in cell-cell adhesion as well as gene transcription. The core of beta-catenin consists of several very characteristic repeats, each approximately 40 amino acids long. These are called the armadillo repeats and have several binding partners. As soon as a new beta-catenin is synthesized, it wants to bind to the ligand that is most accessible. Beta-catenin is involved in 3 different pathways, which will be explained right now. The first pathway is the pathway that beta-catenin encounters towards the formation of adherent junctions. Do you remember the adherent junctions? We are now going to take a better look at the synthesis pathway. After that beta-catenin is synthesized, it meets E-catenin in the endoplasmatic reticulum. The beta-catenin will bind to the C-terminal of the cytoplasmatic domain of E-catenin. This reaction is phosphorylated by the serine-threonine kinase CK2. This interaction is required for the catenin to exit the endoplasmatic reticulum. After this interaction, the beta-catenin/E-catenin complex travels to the basolateral membrane. The beta-catenin also undergoes another interaction. This interaction is with the alpha-catenin, which forms the link between the E-catenin-beta-catenin complex and the actin cytoskeleton. Another function of beta-catenin in this pathway is the prevention of destruction of E-catenin. Without the presence of Wnt, beta-catenin masks the pest region, which is depicted in green. This region is the catenin binding region of catenin and a sequence of peptides which can be phosphorylated and ubiquitinated on the E-catenin. When the Wnt signaling is activated, beta-catenin can't mask the pest region anymore because it is transferred into the nucleus. Now, the pest region of the E-catenin is recognized by the skip-colin F box, ubiquitin E3 ligase, and it will be destructed via proteasomal destruction. Now, let's continue to the second pathway. The second pathway causes destruction of beta-catenin so it can't enter the nucleus to initiate gene transcription. Cells have a special complex dedicated to the destruction of beta-catenin. This complex consists of 5 different proteins: APC, adenomatosis polyposis coli, axin, DVL, disheveled, GSK3B, glycogen synthase kinase 3, and CSNK2A2, casein kinase 1 gamma. In the absence of Wnt, the beta-catenin is recognized by the APC-axin complex. After this, the beta-catenin is transferred to axin. While axin holds the beta-catenin in place, GSK3B and CSNK12 phosphorylate beta-catenin on the serine 45' priming site on the N-terminal. Then, the APC holds the phosphorylated beta-catenin and disposes it so a new beta-catenin can bind. The third step is thus when the phosphorylated beta-catenin dissociates from the complex to undergo multiple rounds of ubicatination by the CSF ubiquitin E3 ligase. The CSF ubiquitin E3 ligase contains 3 core units: the F-box protein, which contributes to the specificity and activity of CSF, the skip1 colon, a horseshoe-shaped complex which is essential in the binding of the F-box, and RBX1, which has a small zinc domain called the ring finger, to which the E2 ubiquitin. The ubiquitin group is attached to the beta-catenin by E2. In total, 3 ubiquitin groups will be attached to the beta-catenin. The last step is when the polyubicinated beta-catenin is recognized by the regulatory PA700 protein. This protein is a subunit of the destruction proteasome. When the protein is bound to the proteasome, it is chopped up into small polypeptides, which can be used in. Now that we have discussed pathway 1 and 2, we will continue to the last and most important pathway. This one allows the beta-catenin into the nucleus to initiate gene transcription for cell proliferation. In this figure, the pathway we just discussed is depicted easier. When the body wants a cell to proliferate, it sends a signal to the cell. This signal is a little protein called Wnt. Wnt binds to the extracellular domain of the Wnt receptor. The Wnt receptor is the frizzled receptor, which has formed a dimer with LPR5 or LPR6, a LDL receptor. Frizzled binds in 2 different ways with Wnt. 1. Via a hydrophobic group which binds to the palmitoyl side chain of Wnt, and 2. Via a chain of hydrophobic amino acid which bind to the index fingers. When Wnt is bound to the receptor, DVL is recruited to the membrane and it activates CSNK1G1, a protein kinase, which results in the phosphorylation of LPR5 and 6. This phosphorylation leads to the recruitment of the destruction complex to the membrane. The destruction complex binds to the receptor complex via a binding between the DIX domain of DVL and the DAX domain of axin. The GSK3B can then phosphorylate the LPR6 tail. The phosphorylated LPR6 tail serves as a second binding place for the axin. Now that axin is firmly attached to the receptor complex, the destruction complex is fully disabled. In this manner, the beta-catenin can't be removed from the cytoplasm, which causes an accumulation of the beta-catenin. Because of this high concentration, loss of beta-catenin will enter the nucleus to initiate gene transcription. Without the presence of Wnt, there's no beta-catenin in the nucleus. The DNA is tightly controlled by a protein called Gaucho, TLE1. TLE1 has a Q domain and a W40 domain. The Q domain of TLE is bound to the TCF7L1, which is attached to the DNA. The WD40 domain of TLE binds to several co-repressors, histone deacetylases. Together, 4 TLE1s form a tetramer, which blocks the access to the DNA for regulatory transcription factors, which is called deacetylation. When the beta-catenin enters the nucleus, it forms a complex with TCF7. This complex replaces the TCF7L1 that was bound to the DNA, which causes the loss of TL1 and its repression partners. This makes the DNA more accessible and it is called acetylation. The TCF7 beta-catenin complex also recruits several co-activators. One of those is CRAB. This histone acetyltransferase binds to the C-terminal of beta-catenin. This makes the promoter region accessible to RNA polymerase pre-initiation transcription complex and other transcription factors. FAB, the signal transduction we just discussed, is disturbed. This is because of a mutation in the APC gene. The APC protein has 6 important regions. Among these are the APC motif region and the APC cysteine-rich motif region, which are both involved in the beta-catenin binding. Then there's the APC basic region, which is a stretch of charged amino acids. Another region is the SAMP motif region, which binds to axin. EB1 region is the end-binding proteins and is associated with microtubule binding. And at last, the armadillo repeats. The mutation in APC is a nonsense mutation which leads to the creation of a premature stop codon and gives rise to a shortened, truncated protein. The mutation lies in the APC motif region and in the APC cysteine-rich motif region. Because of this mutation, beta-catenin can no longer bind to APC, thereby preventing its destruction and causing an accumulation of beta-catenin in the cytoplasm. This will allow beta-catenin to translocate into the nucleus, where path 3 is executed. This causes the activation of lots of different genes, which will lead to a high replication rate, resulting in excessive cell division. This excess of cells gives rise to the benign polyps. The mutation in the APC gene by itself is not enough to develop cancer. This requires a number of other alterations like the mutations in KRAS and P53. We already mentioned, the treatment for FAP is a colonoscopy every 1 to 2 years. If this colonoscopy discovers a malignant polyp, this polyp will be removed via the colonoscopy. When the cancer is already in an advanced stage, then the same treatment as for a normal colon cancer is indicated. When the patient has reached an appropriate psychological age, a preventative colectomy is recommended. Another way to treat FAP is by small protein inhibitors. This type of medicine acts upon the different proteins in the Wnt pathway. This treatment is still experimental and is being tested in different trials. Promising agents are IWR1, which stabilizes axin and promotes the formation of the destruction complex. IWP2, which inhibits acetylation and secretion of Wnt ligands. XAV939, which inhibits degradation of axin and thus promotes the formation of the destruction complex. Now we've come to the end of the video. Thank you for watching. For any questions about the pathway, don't hesitate to comment.